right. Yeah, this is the GWA really is game changing, and Kevin, I couldn't imagine having a better work spouse than you. So, and I do want to recognize Oxford MD Anderson as the sponsor of this track, of this block. So next, I want to introduce our next speaker, Dr. Melyssa Bratton, and she's going to be talking to us about multi-cancer early detection. This is a concept you're going to be hearing more and more about, right? This is the idea of screening for cancer with a blood test. So a liquid colonoscopy, a liquid mammogram, if you will, but not just screening for a few cancers, screening for lots of cancers. And I have no doubt, and I've been wrong with my predictions of the future before, but take it for what it's worth, but I have no doubt that in 10 years, people are not going to be referred for routine screening colonoscopies if you have a normal risk patient. You're not going to be referred for mammograms. You're simply going to go into your annual visit with your doctor and do a blood test like you do for cholesterol or anything else. You're going to do a blood test for cancer. And only if that is positive, then you would go on to more invasive things. So Dr. Bratton is going to discuss this in more detail with us. Dr. Bratton received her PhD from University of Mississippi Medical Center in Jackson. She was at LSU for quite a number of years and has been at Ochsner for several years as well, where she runs our biorepository. In addition to being a traditional biobank, the biorepository is also in charge of running prospective collection studies, blood draw studies, and so on and so forth. And she has a special projects team that operationalizes these studies. I think you have a dozen people or so on the team. And one of the things they do is they operationalize our GRAIL study, our GRAIL studies. We have Eric Fung, who is one of the leaders of GRAIL, who's going to be serving on a panel in a few minutes. And so without further ado, I will hand it over to Melyssa, and I hope your slides.
Yeah, they're on there.
They're on there somewhere? Okay, you know where to find them? Okay, all right, thank you.
Thank you. Let's see. Let me see if I can get out of here. Okay, thanks, everybody. Thank you, Mark. All right. Let me see if I can go to this. Okay, great. All right. I'm Melyssa Bratton, and again, I'm at Ochsner Health. I'm the manager at the biorepository there. Really appreciate the opportunity to get up here and talk to you guys about multi-cancer early detection. I'm going to go really fast because I have way too many slides. This is a really big topic, and I want to talk about it and be equitable about talking about several of the platforms that are available for MCED testing. My direct experience, as Mark said, is with GRAIL and their platform, the Gallery platform, but I'm going to try to give you a history of MCED testing and why we're doing it, how we got there, and then some of the major platforms that are being developed with that technology. I don't have any financial disclosures. So why are we even doing MCED testing? Well, here's one of the main reasons. Because about 70% of the cancers out there do not have any primary screening methods for them. And so they are just incidentally discovered, and only 30% of the cancers have screening methodologies: breast, colorectal, cervical, lung, prostate. And so the unmet need here is to find a way to develop screening technologies for the 70% that don't have them. And so MCED tests, which are blood tests basically, where they can detect multiple different types of cancers just using a blood draw, could potentially fill that gap. And so the critical need for better screening methods also includes, another reason to do this is to detect cancers early on. So one of the main problems in oncology is that patients present with late-stage cancer, and those patients are what's driving mortality in cancer, right? So not only are 70% of these patients not able to be screened, but often they don't present until they're late stage because they don't have any symptoms until, like pancreatic cancer patients, they present, they're symptomatic, they're already late stage. So the MCED tests offer not only a screening mechanism, but also a potential way to predict or find these cancers in their early stages. I'll also say that right now it's important to note that the MCED tests are only meant to be complementary to the screening protocols, not to replace them at this point. And I will talk about the importance of that in a moment. So you all probably know this, but I just wanted to drive this point home, and that is that we all know that if you can detect a cancer in its early stages, the patient has much better outcome. And I'm showing here the ones that several of these already have screening methods, but obviously the pancreatic doesn't. And so if you can, detect these in the blood in stages 1, 2, even 3, you have a much better outcome of patient survival. Now, let's step back, and I'm just going to give you a real brief overview of how we got here. So most of these platforms utilize cell-free DNA or circulating tumor DNA in the blood as a detection method. And this has been around we've known that circulating tumor DNA has been there since the 40s. But it wasn't really leveraged or found to be associated with cancer per se until the late 70s. And then a group in the 80s and 90s were the first ones to say, okay, we can show there's a direct link between this cancer DNA, tumor DNA, and certain types of cancer. Then a really interesting set of studies in the 2010s showed, and this was just incidental findings of non-invasive prenatal testing studies, that some of these moms that were going in prenatal testing had these weird DNA patterns, and they linked it back to all the moms that had these weird DNA patterns actually had cancers. And so this was the first time that they were able to show that you could detect cancers in asymptomatic patients. So at that point, all the light bulbs went off. Hey, we can leverage this kind of these signals to do screening tests in people who are asymptomatic. So that is when MCED testing really, really kicked off. All right, so the current milieu of MCED design basically uses several different types of biology. Most of them use methylation patterns of this DNA that's found in your blood, different methylation patterns. And the methylation patterns the good thing about them is that they're tissue specific. So most of these platforms will offer tissue specificity of each of the signals. In other words, when you get a positive signal, they can tell you, okay, you may have cancer, but also it's probably, lung cancer or breast cancer because of the specific methylation patterns in these fragments. And they will also use fragmentomics, which is the size of the different fragments they can detect, copy number alterations, and sometimes they even incorporate proteomics and somatic mutations into the platforms. So as you might imagine, the FDA is highly regulates these platforms, and what they look for when they are in the approval process of each of these are the following. Obviously, they want to know that they are analytically valid. In other words, are the tests reliable and reproducible? And they really want clinical validity. In other words, prospective performance data, so large prospectively enrolled clinical trials. And in those trials, they really look for sensitivity, specificity, positive predictive value. What that is when you get a positive test, do you actually have cancer? Because sometimes there are false positives, right? So that's a big issue. And also, it's really important the tests have tissue of origin, so that if you have a positive, the test will tell you breast cancer, ovarian cancer, so that you don't just go to your oncologist and say, well, I have cancer, I don't know where it is though. And then they're like, well, let's just do some random testing to see where it is. So if you have a breast cancer, they can do a mammogram, that sort of thing, and start. Another thing to look for is the benefit to risk profile. So are the clinical benefits, do they outweigh the risks of, like, say, for example, false positives? So patients who have false positives they're under a lot of stress, anxiety. Is that mitigated by all of the good results of the people who actually do get the true false positives? and are able to be treated. You have to be really careful when you design your clinical study what your primary objectives are going to be so that they're met. And then the post-market and real-world evidence data, in other words, ongoing evaluation, long-term evaluation of your data, very important. Okay, so I put this up there because what you'll see, and I'm going to show you some data from the different platforms, is I put this up there just to show you that a lot of these trials, the inclusion criteria is that, you have to be at least 50 years old to participate in most of these trials. And that is because the data has shown that once you hit 50, your chances of getting cancer goes way up, which is really terrifying to someone who's over 50. But it's just the way it is. And so that's why I showed that slide. All right, so I'm going to talk today about three different platforms. As Mark said, the Gallery platform is what I'm most familiar with because I've been working on 2 studies, Pathfinder 2 and the REACH study at Ochsner. It is also the platform that is the furthest along in FDA approval submission. And Gallery, GRAIL submitted the Gallery test for PMA in the end of January. So it has the most data, published data, and so we'll be talking a little bit about that. And I'll also talk about Guardant's Shield MCED test and the Exact Sciences Cancer Guard test. I'll briefly mention a couple of others. But there are dozens of MCED platforms out there being, tested. So it's not, it's a really wide open field. Okay, first of all, Pathfinder. Pathfinder was the first prospective interventional study that GRAIL put together, and it enrolled 6,600 asymptomatic adults over 50 using the Gallery test alongside standard screening to assess real-world performance. The overall cancer detection rate was about 1%. The positive predictive value was 43%, which means that of those 1% detections, 43% of those subjects actually had cancer after further workups. Of those, 88% of them, the CSO accuracy was 88%. So the correct tissue prediction. The specificity was 99.5%, so a low false positive rate. That led to the follow-up study, the Pathfinder 2 study, which Ochsner participated as a site. That was a much larger study, and it evaluated the test performance at a larger scale. The study enrolled over 35,000 participants across the country. The specificity was 99.6%, which was 0.4% false positive rate. Positive predictive value was over 60%, and the CSO accuracy was over 90%. And also GRAIL reported the MCED testing increased to screen-detected cancers nearly 7-fold over AB screening and approximately 3-fold over ABC screening, and this highlights MCED's complementary value for cancers without established early detection. That data was released only in October of 2025. So these are all very new data. We will obviously be getting more as the next year. I just want to show also the Pathfinder 2 sensitivity data. So these are and this is important to note that the tests are really trying to pick up early stage cancers. And so you can see that the stage 1 and stage 2 and stage 3 detection levels, they go up as you get higher stage, right? Later stage. And that makes sense because once the tumors get larger, they're going to be spitting out more DNA, and it'll be easier to pick them up. But it's also important to note that you do get a nice level of detection in stage 1, 2, and 3 in these tests. And so I just wanted to put that on there. The 12 cancers that account for the 2/3 of the deaths are on the right-hand side, and that is shown in the detection of those are shown in the dark blue boxes there. You may have all heard of some recent data that was published about GRAIL's NHS Gallery randomized trial in the UK. That was a very large study, and around 140,000 subjects were screened and enrolled into usual care versus usual care plus gallery testing. They just started publishing data from that or giving us some data from that, the primary endpoint. The formal primary endpoint was not met, highlighting the difficulty of demonstrating stage shift. Within a single trial, stage shift meaning does adding the gallery test or these MCED tests into a population on top of standard of care shift the diagnosis of stage 3 or 4 cancers to the left, to where now you see more stage 1 and 2 cancers. Do you catch them before they get to stage 3 and 4? However, so the primary endpoint of that study was that they were going to reduce 3 and 4 cancers. However, the study did show that over 20% year-on-year reduction in stage 4 diagnoses for the 12 lethal cancers. So there was a reduction in stage 4. And so this, the observed stage shift suggests a clinically meaningful impact. And so more time needs to elapse before, they can show us more data on that, but it is promising, I think. And so we'll just see how that plays out. And we're still waiting on Pathfinder data, which we'll get hopefully soon. And I think that's exciting to wait for. Okay. Next, we'll talk about Guardant Shield, so the Guardant is working on its own multi-cancer early detection test. So they have also developed, so they're it's in development, and they're working on a test so the gallery test can find over 50 different types of cancer, 50. This particular test it's in development, and they have done a blinded case control study on 10 different solid tumor types with this platform. It works by identifying epigenomic signatures in plasma, and also detects cancer signal origin. The cohort that they, go back. The cohort that they enrolled, it was 962 participants, 472 cancer cases, 442 non-cancer controls, so it was a controlled study. They knew who had cancer and who didn't. And then you see the median age and the females versus males. So the results of this case control study showed a strong specificity, so 98.6, and an overall sensitivity of about 60%, CSO accuracy 92%, and stage 3 and 4 sensitivity was 81%. So as you go up to in the later stages, you will see an increased sensitivity, and that's just kind of across the board, and that's shown below in the bars below. So over on the right-hand side, I'm showing you some of the top performers. So each cancer subtype is going to perform differently in the test. And so what you see here is that prostate, and this is all of the different platforms. I didn't show you that with Gallery, but all of the platforms have these differences. So prostate, 21% overall, esophageal stomach, 96% overall specificity. So some of the more aggressive cancers do better as far as being detected with these tests versus some of the more indolent cancers like breast and prostate. So takeaways: Guardant Shield, clinical implications and path forward. The study exceeded specificity target. 98.6 was observed when the threshold was 98%. Accurate signal of origin, 92% accuracy. They do have an FDA-approved CRC test already, and so perhaps they can include that with this test to make a single blood draw that may amplify the clinical value of that test if they put them together. So they're now undergoing, I think, a prospective validation, and the next step would be a large prospective study to move forward toward FDA approval. Next, exact sciences. I need to move faster. Exact Sciences has their own test called Cancer Guard, a multi-analyte blood test for broad spectrum cancer detection. They have, I'm going to show you a little bit of data from an ASCEND, from a ASCEND trial, which again is a case controls trial, prospective multi-cancer study, and the difference in the Cancer Guard test from the others is that they also use proteomics in their test in addition to DNA. So it's got two different types of sample analytes. And so they are building this study on the background of a retrospective case control study that they had. The biomarker strategy, again, the single classifier combines methylation and protein signals with machine learning, and they yep, here we go. The cohort structure of 6,000 prospectively collected samples. So they had 1,400 cancer cases, almost 5,000 non-cancer controls over 21 different tumor sites. Here's the training control, the independent test control. Here are the results. So the overall sensitivity was 49.9%. Late stage detection was higher. Early stage sensitivity, 23.6%. Again, it's lower in the early stages, but it goes higher in the late stages. Specificity, 99.0%. And I think I have on the left side the stages broke it down. Okay, here I have a table showing kind of a comparison of all of them. And again, this should be taken kind of with a grain of salt because you can't really compare studies to each other directly, but just to have them sort of, you know, all of the different platforms on one thing, kind of wanted to show that side by side. And I put in the SHIELD study, the CRC FDA-approved platform on there just, you know, as a comparator, even though it's not an MCED test. I won't go through that, but it's just, you know, FYI, this kind of thing. Okay, other MCED platforms. Briefly, want to discuss some other MCED platforms. Interestingly, I just want to talk very briefly about CancerSeek because it has an interesting story. It's not really a current MCED platform, but it's kind of the original platform that all the others are based on. And some guys some people, I won't say guys, some people from Hopkins put this together in 2018, and it eventually turned into the platform used by Exact. And so, it was kind of like the platform of the design that everybody else kind of piggybacked on. Then we have one called the Carcinoma platform. TrueCheck's got a platform. A lot of people have platforms, but they're in the very beginning stages of development. Okay, important stuff. Current limitations, open questions. Right now, none of these studies have proven a mortality benefit, okay? But we just need more time. I mean, the data is still very young, and so we'll have to just observe longitudinal data, see how these patients do long-term to get really good mortality benefit. There is limited early-stage sensitivity, so, and that's, when you're designing these tests, you want to get good sensitivity, but you also want to limit false positives, and so that is going to limit your early-stage sensitivity. Some of these early-stage cancers will be missed, but the third, stage 2 and 3, when you get up to like stage 3, you start getting really good sensitivity. There is a risk of overdiagnosis. So MCEDs may detect indolent cancers, and that would never cause symptoms and might lead to treatments that are unnecessary and provide potential harm to patients. There are false positives, and that which lead to diagnostic cascades, which cost money to hospitals and patients. Health equity concerns. Right now, none of these tests are FDA approved. None of them are approved by insurance. They cost a lot of money, hundreds of dollars. There are people that can't afford that, and those people are often marginalized as it is, so there are equity concerns. Risk of false assurance. This is a really big one, okay? People who have a negative result often feel like they don't have to get their screenings. And I can speak to this personally because I enrolled both my parents into this REACH study. They both got negative results, and my mom's like, whoop, I don't have to get that colonoscopy. After I told her and counseled her over and over again, okay? And I'm like, absolutely not. That is not what that means. So a lot of work's going to have to go into counseling patients on following up with their regular screenings. What's the road ahead? A Medicare coverage pathway. So the good news is that in February, the Nancy Gardner Sewell Act was signed into law, which established a Medicare coverage pathway for FDA-approved MCED tests, which should start covering, Medicare should start covering this test in 2028, and hopefully that will lead to full insurance coverage in the future. The FDA is developing a Class 3 pre-market approval pathway for MCED tests. The NCI Vanguard study is a really large study which launched last year to evaluate MCED feasibility and diverse average risk adults. Mortality endpoint trials. Again, we need to know if this is going to affect mortality of patients. And then next-gen assays, it's just happening so quickly, including multi-omic integration, AI-driven. AI is going to change a lot of things, as we all know from this trial, from this talk. Okay. Wow, I'm way past. Okay, that's it. Thank you.